Phylum Amoebozoa, amoebas, Entamoeba histolytica. I said a few things about Entamoeba histolytica that they cause dysentery. Dysentery is bloody diarrhea. Bloody diarrhea, and it's devastating. It can kill. Again, in the United States, people who become infected with it, not much of a problem because there is medication available. The medicine is more advanced, more available. Not only advanced, it's more available than other parts of the world. So we can treat it in the United States. In younger children, it can kill adults. It depends. You have other diseases or not. When we say in other countries, one person is not only infected with one parasite. They can have 4 or 5 parasites. Okay, so it can be devastating. Entamoeba coli does not cause much problem. But again, if you look at most of parasitology textbooks, they classify it as a parasite. It's not a commensal. It's a parasite. They refer it as a parasite. Entamoeba gingivalis, another amoeba which I talked about it. This is a commensal. It is found in the mouth. The way we get it from by kissing. You kiss another person, you get Entamoeba gingivalis. I'm not saying everybody has it. Most everybody have it. So don't say I'm not going to kiss you because we passed Valentine's. Is that right? Because you might have Entamoeba gingivalis, and it's a commensal. Phylum, again, under phylum amoeba, they are not under phylum Amoebozoa. This is another phylum by itself, phylum Foraminifera. We do have a slide of it. They have a test. They call it a test. So the amoeba is inside of that test or shell. Test, T-E-S-T, shell. Same thing. So the amoeba is inside, and what you're seeing right now, you're on the slides, you are seeing the shell or the test of the amoeba that resided inside of the shell or test. Made up of calcium carbonate, silt, silicon, so on and so forth. The other phylum is phylum Radiolaria. On the microscope, you should be able to tell them apart. We do not have holozoans, but we do have, the slides say Radiolarian on your slides or Foraminifera on slides. You look at the shells, they're shaped differently. But you should, like Foraminifera, you have 4, 5, 10 different, on your slides, different shells, different type of shells. Same as Radiolarians. But again, you look at them, they're distinguishable. The two are distinguishable. Phylum Dinoflagellata, Serratium. You do have slide of serratium, look like a star, look like a, you looked at it, look like a neuron, but does not have axon. It's a single cell organism. And then they have flagella, and they do have, what else? They do photosynthesis, okay? Free living, all of these are free living except these guys. They're phylum Amoebozoa, the ones that I mentioned. There are free-living organisms, the amoeba in phylum Amoebozoa, they're free livings, but these guys are either commensal or parasite. I hope I'm making some sense. But these guys, all of these guys are free living. We do not have any parasitic ones on these phylum. Here are the amoeba. Here is Foraminifera, Radiolaria, and holozoans. We don't have holozoans, but that's what they look like. You might think this is a snail shell. It is not. Okay, this is your Radiolarians, Foraminifera. It looks like a snail shell, but it's not on a microscope. Okay, phylum Heterolobisia, Nuclearia fara. We touched up on it today, this morning. This is a free-living organism, but when we human ingest it, it becomes parasitic. And devastation, brings along devastation. The name of the disease that this parasite caused is called primary amoebic meningoencephalitis. The abbreviation for it is PAM, P-A-M. During lab practical exam or lecture exam, I hope you don't write down PAM. You can write down PAM, but after you write this first. That's how they do it in scientific paper. They write it first time, the whole thing, And then after that, they refer to the disease as PAM, PAM, PAM. But first time, we have to spell it out. Okay, so primary amoebic meningoencephalitis. Flagellated and amoeboid trophozoites, binary fission. They multiply by binary fission, means asexual reproduction. And transmission by amoeboid form, death within 5 to 7 days. So he will kill their host quickly, rather quickly. Doesn't wait around. And it's very hard to diagnose. Most of the time, when they find out the cause of the death was this parasite, it's in autopsy. When the patient is dead, so they open up the patient, and they find the amoeba in the brain. Of course, they give it to pathology. You have to find it on a microscope. With naked eye, you cannot find them. And then, of course, intravenous, the drugs, there are drugs available, but the doctor has to identify it and find out. The patient comes to the doctor and say, I have headache. And the doctor says, go home with some aspirin, ibuprofen. That's not cured, okay? But if the doctor asks the right question, have you been swimming? Have you been in a dirty kitchen or somewhere that you ingested the water? I have examples of it for you guys. Then it can be treated. It can be treated by these drugs, and these are mostly are. Here is a person swimming, and here is the transformation from amoeboid to flagellated one, to amoeboid one to cyst one and amoeboid one, and of course the amoeboid one in human cause a lot of this. Here are the organisms. Here is the amoeba. Right here, they are devastating. They're killing the cells. And of course, there are signs on the beaches sometimes. They say keep out because of amoebic meningitis. Right here, that's what they're referring to, Naegleria fowleri. Here is the one that you do have in the Naegleria. You do have in 2x2 slides for your lab practical purposes. So in the lab practical exam, you might see this and ask you what is the name of the organism Naegleria. How to get Naegleria in ponds and lakes, water into the nasal passages, immersions in bath water, like as I said it in baptism. It's been reported, and some people got it during baptism. Playing in warm, muddy puddles after rain, usually. It's not in cold weather. This parasite, people ingest it in warm, hot days. Usually, unless it's indoor inside of a kitchen, place it through a water school playground. Of course, in some baptism, as I mentioned, that is possible. Okay, the other phylum, next phylum is phylum Viridoplantae, which is your volvox. Chlamydomonas, of course, is one of them. The other one is volvox. Both of them are underlined. Both of them is the name of a genus. Look at capital C, capital V. It means genus. I did not give you any species on these. Volvox, it has a division of labor. What I mean by division of labor, they have certain cells which they do photosynthesis. It is responsible for providing food for the colony. And other cells, they undergo sexual reproductions, which is for reproduction purposes. Macrogametes loaded with food, and microgametes forms sperm and egg. Zygotes multiply and break away, and here's a life cycle of it. So the slides you have on the microscope, do not worry about finding sperm and egg on your slides, but you should find out on your colony. This is the mother colony. Inside of the mother colony, you have daughter colonies. These colonies will get out of the mother colony and form their own colony, and eventually inside of them you do have daughter colony growth. So this would be asexual reproduction of the volvox. They are flagellated organisms. In the exam, sometimes I ask, and you write down they're ciliated. No, they're flagellated. They have flagella. And then this is the sexual reproduction, which the sperm swims and fertilizes the egg inside of these colonies, and they form zygote, and zygote sometimes get out or inside of the colony develops to another colony, so that's what they call it. So this is one example of a multicellular organism, of multicellular Protista. So far, whatever I talked about, as far as Protista goes, they're unicellular, except this one. This one is a multicellular, and it's a colony of cells, okay? And the cells, when they do photosynthesis, they are called vegetative cells. And the other cells, sperm and egg, they are reproductive cells. Phylum Diplomonata, Golgi bodies and mitochondria are lacking. And one is, we talked about it today, is Giardia. Giardia lamblia, there are two different types of Giardia, Giardia lamblia, Giardia intestinalis. And here's the life cycle of it a little bit. Here is a Giardia trophozoite stage with flagella all around them. 1, 2, 3, 4, 5, 6, 7, 8. I wrote on the board 4. So anywhere they have those, but they have two nuclei, the trophozoite, the feeding stage in the intestine. In our intestine or animals' intestine, you find that. And then in the poop, in the feces of their host, you find this. This is the cyst stage. We do have slide of both of them. You have slide of trophozoite, and you do have slide of the cyst stage, both of them. We have both of them in the lab, so make sure you look at it. Then what will happen, let's go over this. Giardia lamblia, flagellated ones, trophozoite in the large intestine multiply asexually and by binary fission, and then diagnostic and infective stage, you do have it in a resident infective stage, cyst passing feces, trophozoites by found in commonly in sort of fluid feces, and then what you have here is a method of infection. Man ingests infective cysts transmitted by feces, fingers, food, fomites. Fomites are inanimate objects. Inanimate objects like such as your pen, pencil, key, your cell phone, sometimes you put it in your hand. Whatever that is not a living object, and you put it in your mouth, and it has these cysts on it. They have the cyst on it. And flies. How flies do this? They fly sit on the feces, and they grab these cysts on the leg, and they fly, and they sit on somebody else's food or water. Then that person eats that food or water, voila, you got Giardia. Okay, so that's what I mean by cysts, by flies. Again, don't think this is a direct life cycle. Still, this is an indirect life cycle. But the fly, on the leg, they grab these cysts from a feces, from diarrhea, and they go sit on somebody else's food. Still considered to be in direct life cycle. Sorry. Still, it is a direct life cycle. Cyst pass in the small intestine, excistation, remember that? Excisment occurs, and Giardia, the life cycle goes on and on. Okay, phylum Paramecium, large Golgis, opposite of the last phylum we studied. Giant amoeba, you might think this is an amoeba, but no, because of DNA, we talked about this, because of DNA, they decided to put this in a different phylum. Giant amoeba. Trichomonas, you do have slide of it. Trichomonas vaginalis. And also Trichonympha. Trichonympha, here I did not give you the name of the genus, but the name of the genus is there. Trichomonas vaginalis, you should know that. And it's the only one that is transmitted, the only one that we know in this class that is transmitted by sexual contact. Okay? Trichonympha is an example of what? Remember that this is, giant amoeba is also a parasite. I'm not going to talk about it too much, but Trichomonas is sexually transmitted, and in case of people who get it female, they have foul smell, vaginal discharge. These are signs and symptoms in female. In males, usually you do not have much of a problem unless the numbers are high. If the numbers are high, same thing, that's now maybe some discharge from the penis, so on and so forth. And again, female get it from male. That's how it is. It's flagellated. You do have flagella, Trichomonas vaginalis. Okay, trichonympha is an example of what? Commensalism, mutualism, parasitism.
Mutualism?
Mutualism. Mutualism. And it's found in the gut of the termites. When termites eat wood, then the wood is broken down by these organisms to simple sugar molecules. So the termite can use the sugar, and also the trichonympha can use it. There are different species of trichonympha in the gut of the termites, but I only mentioned one of them, so you should know trichonympha. And I did not give you the name of the species. I gave you the name of the genus. So these are all Parabasalia, based on your textbook. Flagellated protozoa that cause African sleeping sickness, euglena. In old, they are both, this is Trichomonas vaginalis, again, in a different phylum than Trypanosoma. But anyhow, in old days, old days, when I was a student, both of these were in the same phylum. In old days, they classify them based on the locomotion, like their DNA, but anyhow. Okay, phylum Apicomplexa, all of the members of this phylum are parasitic. I have not said that about any other phylum. I have not said that about any other phylum. This is the only phylum that all of its members are parasitic. They have specialized organelle. Specialized organelle, it means not too many species or not too many organisms on planet Earth have this. And it's called apical complex. Okay? So the only one that comes to my mind so far that we studied was kinetosome in euglena or in Trypanosoma. But other organisms we studied, they did not have any specialized organelles. Maybe Paramecium has ampulla, not the contractile vacuole. That's not specialized organelles. A lot of organisms have that, but in Paramecium, you remember that? Ampulla, that is also considered as a specialized organelle. Okay, so apical complex is membrane bounded. It has a phospholipid. That's what the definition of organelle is. You have a phospholipid membrane around that structure. So they do have phospholipid around it, and inside of that phospholipid membrane bounded, they have a structure called rope tree. Rope tree, and I only gave you two of them, micronemes. There are two structures that they help the parasite to gain entry into the host cell. So if you have a host cell here, for example, host cell, and then you have this parasite here, so they have this organelle, they release some kind of enzymes or molecules to digest away the cell membrane, so the parasite can enter the host cell. That's what the function of apical complex is, to gain entry into the host cell, and of course cause damage later on. They have some stages you will see in case of malaria. In case of malaria, they have some pseudopod intracellular stages, flagella in gametes of some species, and asexual and sexual reproduction both. And some requires intermediate host, like I erased it, but like in case of malaria, mosquito is an intermediate host, or the intermediate host is not a good example. Intermediate host, that's where asexual reproduction occurs. In case of malaria, human is the intermediate host. The final host is mosquito. And I'll go over that. I will go over that. Hang on. Okay, so intermediate host pretty much means where asexual reproduction occurs. That's what intermediate host. Where the sexual reproduction occurs, final host. They call it final host. Okay, and some develop oocysts or spores, and I will talk about that. Oocyst is equivalent to zygote when microgamete and macrogamete come together and form the oocyst. And the oocyst sometimes is in the feces. It depends on the parasite, on the organism we talk about. Sometimes it's inside of the mosquito gut in case of malaria. It depends what organism you're talking about. Okay, class, again, for the lab purposes or lecture purposes, because there's so much phylum you have to know, I will not ask you to know the name of the class Coccidia, the most devastating protozoa, Apicomplexa, are in this class. Most important class, again, because they have a lot of devastating organisms to human are in this class. Pyroplasma in cattle, Texas red water fever caused by Babesia bigemina. We human have it. Again, in human, sometimes it's misdiagnosed with malaria. The people in the lab miss it on the microscope. They think the person has malaria. The doctor prescribed the wrong medication. So the signs and symptoms are almost same. Another organism is Cryptosporidium. Cryptosporidium causes diarrhea by contaminated water, mainly. Fomites, sure. Flies. Remember, they fly from feces and then they take it to somebody's food. So the life cycle here is indirect or direct. Which one? Remember, indirect means an intermediate. It needs a vector, a biological vector. Does Cryptosporidium need a biological vector? No. So it's a direct life cycle. It's a direct life cycle. Coccidia, again, intercellular, Eimeria, Isospora. We do have slide of this one. You do have slide of Isospora, but you do not have Eimeria. And Isospora in AIDS patients can be devastating. Again, Eimeria tenale in young foal chickens. Reproduction of Eimeria, I think we talked about this. Schizogeny to gametes, formation of gamete, fertilization, and out into the oocyst. The oocyst goes in the environment within the feces with fecal matter. It goes into the environment, and other chicks comes in, other healthy chicks comes in and ingest that oocyst in the intestine of the healthy chick. The feces have the oocyst, undergo sporogeny. Well, in the environment, sporogeny can go on, and then the oocyst becomes 8 sporozoites, and outside of the host, again, I guess I thought my next one is that this is explaining what happens in the oocyst. Sporogeny, and new host ingest the sporulated oocyst, sporulated with 8 sporozoites. It comes out of the egg and becomes thousands and millions and destroy the epithelial cells of the intestine. So the chick cannot absorb any food, they get diarrhea, and eventually they will die. Toxoplasma gondii, I will talk about it next time. Give me a rain check on that one. In AIDS patients, you and I might have it, we have no problem, but in AIDS patients, it can be devastating. Definitive host or final host, another name for final host is definitive host. So you have intermediate host where asexual reproduction occurs. Definitive host or final host, that's where sexual reproduction occurs. Sexual reproduction. Possible intermediate hosts are human, pigs, chicken, any animal. Any animal, almost any animal, can be the intermediate host. Plasmodium, malaria transmitted by mosquito, which the name of the mosquito you should know is Anopheles, female mosquito Anopheles. And then sporozoites injected into human by insects, sporozoites in the liver, initiates schizogeny. I think I'll show you the picture, and it makes since Plasmodium falciparum, there are 4 different types of Plasmodium. Plasmodium is the name of the genus of malaria. The reason they call it malaria, Roman soldiers used to go fight in remote area in Africa, Asia, other parts of the world, and they used to come back to Rome, and they used to come down with this sickness. Of course, during that time, nobody knew what it was. But they call it mal, it means bad, area, air, it means air. So these soldiers went to these areas, and they came back to Europe, in Rome, and they had these sicknesses, so the soldiers they ingested malaria. That's how the term malaria came about. But later on, when they studied it, say 100, 150 years ago, they saw these organisms, Plasmodium, on the microscope. Okay, so they call it, we cannot call it malaria, it's called Plasmodium. And there are different types of Plasmodium. Plasmodium falciparum, which is the most devastating one, kills. We have Plasmodium vivax, we have Plasmodium and Plasmodium malariae. Okay, so this is the most devastating one. And penetrates red blood cells, and then of course it will kill. In red blood cells, trophozoites feeds on hemoglobin. Sexual reproduction, of course, it happens. Makes microgametes and macrogametes, and mosquito ingests the gametes, and fertilization oocysts. Here is the one that I want you to know. Are you ready for this one? Let's go. Okay, so let's go with the mosquito, the Anopheles. Take a blood meal from human and ingesting saliva into the human. In the saliva of the mosquito, not the blood that they transmitted to the human, the saliva that they transmit to human, you have sporozoites. And I will talk about where sporozoites come from in a minute. Hang on. So those sporozoites by our blood is carried to where? Good old what? Liver. Liver filters everything, right? We talked about that. Detoxifies, filters. And when they get into the hepatocytes, this is hepatocytes, sporozoites enter liver cells and undergo schizogeny. What does schizogeny mean? So you remember I gave you that terminology before. So you have a liver cells, a hepatocytes with a nucleus in here. One Plasmodium enters the cell, right? And it has a membrane around it too, right? Am I making some sense? It has Apicomplexa. We talked about that. And right here, inside of that cell, you have nuclear division. What follows nuclear division in Bio 1 you learn? Cytokinesis. Does cytokinesis occur here? No. Just inside of one cell, you have the nucleus dividing and dividing and dividing and dividing without cytokinesis. That process is called schizogeny. It's not called mitosis, right? Because mitosis follows cytokinesis. So what shall we call it now? Let's say call it schizogeny. Schizo, it means spill. Schizocilus, you remember that? Splitting. So here are the nucleus of the original organism multiplying. Then they break out and they go invade another cell. Keeps going on. Okay, so you have, and eventually it will get out, maybe one generation in liver or 2 generations in the liver. They get out of liver and they go to the red blood cells and destroy the red blood cells. When they destroy red blood cells, that's when a person feels shivering, shaking, signs and symptoms, sweating, fever. That's when the red blood cells are being destroyed. A tremendous amount of them. Body does not know how to handle them. Okay, so red blood cells are destroyed and merozoites are released. So merozoites, some of them become microgametes, some of them, not all of them, and some of them become macrogametes. Okay, micro and macro. Micro usually in biology are the sperm, and macro is usually the egg, right? So microgametes and macrogametes, and of course you have trophozoites, they feed on red blood cells. Then another mosquito comes, take a blood meal from a sick person, and of course, in that blood meal, you have microgametes and macrogametes. They go inside of the mosquito gut. This is the inside of mosquito gut. And the gametocytes, the microgamete and macrogametes come together, form, the male gametes becomes flagellated. You remember that? We talked about it. And then they come together, fertilization, oocyte, and oocyte eventually becomes oocyst. Inside of those oocysts, sporogeny goes on. So you have one cell inside of it. So what would be? So you have the oocyst, oocyst. You have one nucleus right here, but that one nucleus inside of the oocyst become 2, 3, 4, 5, 6, 7, 8. Usually, in most species, in Cryptosporidium is only 4. It depends on the species. And then in here, they do form a membrane around them. That's why this is called sporogeny. In schizogeny, they don't form membrane around them until this breaks open. When it breaks open, then membrane is formed around it. When they break from schizogeny stage. Here, they have membrane, and then when this gets inside of the host, next host, in case of mosquito, it breaks open. Okay, in some species, this will get inside of the gut, and then it breaks open. In case of like Toxoplasma gondii or Cryptosporidium, okay. But in this case, it breaks open, and they are calling it sporozoites developing oocysts and release. And my salivary glands, they are ready now, and they are injected into human. These sporozoites individually are injected into human, and one of a healthy human, and the cycle goes on and on from one human to the next human. I hope you understand this. Study it. Make sure you know it, because I might ask multiple choice question or essay question before. Yes, Ariana, you had a question.
I have what happens after it multiplies in a good mosquito?
Right here, they multiply inside of the oocyst, right? Oocysts usually have one nucleus here. If you look at it, it has one nucleus. And then inside, inside of the oocyst, they multiply to a fixed number, okay? 8 usually is the magic number. In case of Cryptosporidium, it's 4. But inside of the oocyst, then they come out of the oocyst, and they're individually now. When mosquito bite a human being in the saliva of mosquito, then these sporozoites get inside of human. Does it make sense?
Yeah.
These are the ones that they were from end product of sexual reproductions in the gut of the mosquito. So mosquito in here is a definitive host or intermediate host? It's a definitive host. Sexual reproduction is occurring here. So mosquito is definitive host. Human, where asexual reproduction is occurring, asexual cycle, that's intermediate host. Human is intermediate host, mosquito is definitive host. Eduardo.
So just taking is that just caused because the red blood cells are being destroyed?
Right. That's the main one. That's the problem. If we can figure out how to stop that, I think we beat malaria. And of course, there is no vaccine, you guys. Are you listening? There is no vaccine for an eukaryotic cell. We have vaccine for viruses. We have vaccine for some viruses, of course, I should say. We have vaccine for some bacteria, but we have no vaccine for any eukaryotic cell yet. If they break it, if they break the code, I should say, Da Vinci Code, is that one? There was a movie. If they break the code and they come up with a vaccine, so malaria hopefully will be the first one. Maybe they should work on some of the organisms and come up with a vaccine for other organisms and eventually tackle malaria. But then, what do I? Okay, Cryptosporidium parvum live in small intestine, respiratory epithelium, direct life cycle, not like malaria. Malaria is indirect life cycle because you need the mosquito. Microgametes, oocysts in soil or water, and it can become airborne. Outbreaks in Washington, D.C. and Milwaukee. I talked about all. This is just for you. One more time. Phylum Ciliophora. Let's finish up everything today so we can talk about Toxoplasma gondii on Thursday. Mostly free living, parasitic, some of them. Which is, you do have side of it, parasitic ones, which is Balantidium coli, some commensalism and some colonial, some sessile, Sartorians. The sessile ones is called Sartorians. Larger than the rest of other protozoa. Compared during lab practical exam, a lot of students make mistake between Paramecium and Euglena. Euglena and Paramecium, they do look alike, but Euglena is much smaller than Paramecium. Paramecium is a little bit bigger. Compare it. During lab exam, you can tilt your head, see if it's a 4x or 40x. You can do that. You cannot change magnification, you cannot move the slide, but you can tilt your head, see if it's a 4x or 10x. Okay? They do look alike on the microscope, so don't. They are multinucleated, macronucleus. Undulating membrane cilia are fused into a sheet. Cytostome, stone, it means what? Mouth. They have a mouth, call it oral groove, cytostome. Oral groove is before cytostome. Two contractile vacuole system. We talked about that before. Passage of water and food in paramecium, oral groove, cytostome, cytopharynx, food vacuoles, indigestible out by cytoplasm. Binary fission is possible, as Larry said it, so there are not too many of them. If a disease comes or some kind of natural disaster comes, divide mitotically and macronucleus elongates and divide amitotically. A means not by mitosis, mitotically mitosis. Conjugation. Today I will put out 2 slides for you guys in the back on demo, and today is going to be available only. It's going to be conjugation and binary fission. Make sure you look at those 2 slides because you might see them again on February 27th. Autogamy is self-fertilization. I said when I talk about paramecium later on, I will identify that term,